Okay, so I will start saying a few words about what happened from the moment you do your data submission form, like Rob showed, and then how from that moment what you need to do to actually start creating a new data set. So typically, like we show, you fill in your data submission form that gets over to the hub. Once what happens once that is admitted, in that form you have given us information about your research and the grant ID and who are the PIs and stuff like that. So with that information, we do two things. One is we create a group in Globus that will control the access to your data and the access to your group. And we create a project page in our database that you can use as a sort of the entry page to see all your data set and all your data and all the information about your project. Again, we think about the word project as something that sort of is more at the organizational level. So who are the scientists, who's the grant ID, what's the main focus of the research under that project, and then the actual pieces of data, we call them data sets. So typically what happens once your request is approved, you get an email from us saying that your new project has been created, and to go to this Globus link to join that group that will allow you to start creating data. So in this case, I will assume that this project is the one that we use for demo. So I will try to show you that. So I will go to my copy the URL for that project, go to my browser, paste that link, and that will take me to a page where I need to authenticate. So again, you can choose to authenticate using your institution or organizational login or your Globus ID if you already have one, or you can create one. I will do this following el Globus ID. So once you go through the login, you will end up in a page that gives you the option to join group. So once you click that join group, that we receive a request saying that you are applying to join the group. We accept that group, and then from then on, you can authenticate into FaceBase and directly access your project. So now I will start again. I will pretend that already it's been approved, and I will try to start creating a data set in FaceBase. First, I want to say that I think this is something that Rob has already shown, but I want to show again that again, our documentation page under submit, how to submit data, and this link to our FaceBase curation page basically is extremely useful. So I'm going to demonstrate this, but in any moment, if you have doubts or you don't know how to do something, you should visit this page, and we have a pretty good step-by-step information on how to do pretty much every task. And if you, for some reason, still need more help, you can always send us an email. But I want to make sure that all the key concepts, the data submission process, how to create a data set, how to create an experiment, biosample, replicates, how to upload data, everything is very well explained here. I'm not going to go through this documentation page now in detail. We'll try to basically demonstrate this as we go along. So again, I'm going to go back to FaceBase. Typically, the entry point when you're starting to create new data sets is to try to navigate to your project, and then from your project, they start creating the data sets. So I will go to the project page. I will try to find my project. Okay, I know like my project is called demo for this demonstration, but most likely in your case, it will have a more scientific name. And I don't find the demo project right now. And the reason that I don't find the demo project right now, and typically probably that will be the case for your new project, is because when projects are created originally by default, they're not public. Has not been released, so they're not accessible by anyone other than people that belong to that group. So the reason you do not see it listed right here is because you haven't logged in. So I will log in. I will log in using the ID that I know it's already been added to that group. And now if I look for the demo project, I should be able to see the face-based demo project listed, and I can go and visit my project page. So again, this is mostly something that has information at the organizational level. So it has a DOI and a record ID. The DOI is something you can use to cite data the same way you can cite individual pieces of data, or you can actually cite your whole project if let's say in a paper or anywhere you want to say, okay, this is where I have all my data sets in FaceBase. You can, instead of listing all the individual DOIs for each of the data sets, you can also give a reference to the whole project. Then here in the abstracts, it will be the information that you have. Initially, it will be the information that you submitted that you included when you submitted the data request form. So we copy that data, that piece of document in here, but then you can always go and edit everything that is here. That all these fields are editable. So you can click edit here and modify your abstract, for example. And again, for example, the abstract field in the project, it's a markdown field, so you can be sophisticated. You can add images, links, lists, and so on, right? Whatever. So, and you add those changes. Same thing with the PIs. Initially, we add the people that you listed as PIs in your request form. But if there are other people in your project, most likely, your postdocs or students or other collaborators, you can go here and add them. You click add record, and if that person already has been in your project, let's say Yan Chai, no, Yan Chai is already a PI, but let's say, I don't know, Bruce, I don't know, it's also part of your team, so you can select him there, or if it's a new person, a new scientist that has never been in FaceBase, then you can go create record and create a new entry and add it as a project member. Then the funding, there will be the grant ID. Typically, that's the grant ID that you provide in the form. That here, this is a demo project, so we didn't add anything. You can add list of publications, and you can also add protocols at the project level. The same way you can add it to the experiment level, like Wu shown. You can also go and add protocols at the project level. Then say, and then you can use them. One thing that I want to again say is that once you enter a new protocol, the way that Wu did, protocols also by default are not public. So, but once they are released, anyone in Facebook can access them. So actually, this is not what I want to do, sorry. Yeah, so so you can create a new protocol by creating, adding a new protocol, or you can just pick one that you have used before. So we'll see that later more. And then also once another field that we expose here in the project is the URL to request membership to that project. So if that once this project becomes public, in principle, you can share that URL and people can come here and request membership. And then we go to, there are two sections after that. One is the QC issues. That's something we're going to talk a little bit later. And the data set. So the QC issue is that there are the quality control rules. We have a set of rules that we monitor for each data set. And once the data sets have been created and are in the curation process, we check for any issues that can be missing, like missing experiments or missing replicates or missing protocols. And we include that here. So you will see listed per data set all the issues that we identify, and that you can use that to correct and improve the curation of your data set. So now let's go to the creation and actual data set. So now under your project, you will see listed all the data sets that you have created. For example, in this case, I already created one data set that is pretty much the same as the one I'm going to create now, but that we want you to use as a reference if you are interested in doing this self-guided tutorial. We have a wiki page with all the steps that you can go through in order to recreate this data set, and we have a zip file that has basically pretty much all the data. So in principle, you should be able to do everything that I'm doing now by following this, the structure of this data set and using the data that we provide. Well, now I will imagine that I want to create my new data set. So I will click Add Record and start the process of creating my data set. So one thing that people typically try to keep in mind when we go to create a new data set is, what are the pieces of information that I will need in order to create a data set? And one is that sort of the high level information of data set, and then we go down to the actual structure of the data set, basically how many biosamples do I have in this data, what experiments did I conducted on those on those samples, and what pieces of data go to each of these combination of experiments and data sets. So, but we'll start with the sort of high level description of the data set. And since we already started this data set from your project, then your project information is already populated here. Next is the accession number that is already automatically generated by the site. And then we go to the title. And we try this title to be as informative as possible, because this is one of the searchable fields. Anyone, when you go to the list of all data sets, you can, on the top search bar, you can type anything, and the title is one of the fields that's going to be hit by that search. So if we would like this to be as descriptive as possible so that people can find your data. So I'm obviously not going to be very descriptive now, but this is now, you got to keep in mind that this is like the title, the same way of the title of a paper or the title of any publication that you do. You want to be as descriptive and informative of how the characteristic of your data. So then we have two main fields, and I think that, for example, in the data set that Vu showed, they were very well populated. One is a description that basically, goes through, okay, in this data set, we have data about mouse at these different stages, and we conducted this type of experiments, and blah, blah, blah. Again, we want to be as descriptive as possible. And same thing with the study design. Study design can be in a way, it gives you like a vision of how the whole data set and experiments were envisioned, and they can be like a small abstracts of the protocols, or because many times a data set will include different techniques, so you can say we did this and we did that, and then but it doesn't have to be as descriptive as the protocol, but it can something that someone going to the data set have a first view of what's been done in this, or what's represented by this data. And then the next field is whether this is human subject data or not. So this is very important, because obviously we never want anyone to upload any type of data that can be protected data. And this is basically, in a way, your responsibility, because you need to be absolutely sure that no data that you are uploading here can be human or identifiable human data. So if it's protected data, if data that has any kind of requirements, then you should indicate it here and then contact us, and we will guide you on how you provide that data to us, because that data cannot be added to the site. You can still create the data set, and you can still add the description of the data set. You can still give us all the description about your biosamples and experiments and protocols and all that. That's fine, and that's going to live here in the catalog. But the data itself will be provided to us through a different mechanism, where we basically have asked you to encrypt the data in a given way and transfer it to us, and we store it in our dedicated servers. The last field is whether you want to get a DOI for this data set or not. So typically you say yes. Now, most of the time there are no reasons for saying no. So I click save, and so that has started my data set. I have entered the most basic information. One thing I want to call the attention here in the page, as you can see, we have a lot of fields and a lot of different sections. And sometimes it starts to get confusing as you're working with this. So we have here at the top one button that says hide empty section. So you can actually toggle this button, and it will just hide from your view anything that's empty and only show you what's been entered so far. So that's typically very useful as you go through the process of creating a data set, because then it kind of clears out the clutter. Okay, so the next section are the contributors, publications, protocols, and related data sets. Those are sort of fields that also add additional information about the data set. So contributors, we have a nightly process that populates the contributor field with anyone that is a member of the project. But after I create this data set, next time you come to it, you will have the name of the PIs and anyone that you have added as a project member. But if you happen to add people that are not specifically part of your project, but you want to add them as contributors because they work on the data, you can go here to add record and add a new people as contributors. Same thing as publications. If this data is associated to something that's already been published, you can click add record and enter the PubMed ID for that publication. And now when you go back, when you navigate, so typically every time we click on this, it opens a new tab, but then you can go back to the previous tab to go back to the data set. So, and you can probably close this one, you don't need that one, so you can go back. So, and you can add more than one. You can click add record again, or, so if there's multiple publications associated with that. Protocols section, as you can see, I don't have the add record here because it will be automatically populated by any protocol that gets later added to the experiments. The next field that is interesting and important, and I think that Fu will show an example, is the related data sets. And this is the mechanism that we have in order to create a relationship between the data that you are uploading here and data that can be either available somewhere else in some other repository or internally in Facebase. So you can add a new record, and you can select the type of relationship that your data set has to the other data set. So, and the possibilities are, it can be a geo data set, it can be a geo GDS or geo GSE, it can be a dbGaP data set. So let's say that part of your data, this data has already been uploaded to dbGaP, then you can make that, you can indicate that in here. Or maybe this can be just related to another data set that you previously created in Facebase, that probably were, for some reason, they share something in common. So you can make that relationship explicit in here, or you can indicate that this data is derived from or is secondary analysis of, or is the primary data source of. So we have some cases where some data sets that are just, a computational analysis of data that's been downloaded from Facebase. So you will here indicate that this is secondary analysis of data in Facebase. But let's assume that this is related to some other data set I have already created. So in Facebase, so here, instead of entering the related data set URL, I will actually pick the actual data set that already exists. Let's say that this is the data set that is related to this other data set. So I go there and select it. And then imagine that something I want to start showing here is that you can add more than one of these relationships at once. For example, if you were to have a relationship to a related data set and also to data in dbGaP, we have a very convenient way of sort of rapidly creating these entries by using this clone mechanism. So here on the top right side, you will see the quantity and the clone button, and you can express how many times you want to clone this entry. So I will click 1. So now I have 2 identical copies. I just go and make changes that I need. So in this case, I want to add some reference to dbGaP. Right, wrong. So I'll try to put something as wrong as possible, and then click save. And then I navigate back to my data set page, and now I have these 2 relationships between this data set and other data. The next two fields, like protected human subject and data use limitations, those are mostly related to human subject data. So I will not go through that now. So then the next block of fields is mostly what we call the high-level tags, which are sort of the metadata information that tell us, the characteristics of this data set. And they're very useful when we try to search for data. We have all this, like Rob said, we can use sophisticated faceting search in order to find data. And the way that the faceting works is by going through these tags. And all these tags, most of these tags are based on what we call control vocabularies. So we don't want to have people entering, any type of free text. For most of these dimensions, we have this set of control vocabularies, and whenever possible, and for most of them, we take these control vocabularies for known ontologies. So there are a few ones that are very difficult to do that. So they are more of sort of our own internal control vocabularies and terms that we can enter, but most of them actually come from some well-known ontology. So the first one very important is experiment type. So I will click a record and say, okay, in this data set, I will assume that I did some micro-CT analysis and some RNA-seq analysis of my samples. So I will here search for micro-CT. And then I will click here again and search for RNA-seq assay. So next one is species, right? So I assume that in this experiment, all my samples were mouse. So I will enter here mouse. And we will see that many of these information need to be entered again when we create experiments or the biosample, but here it is sort of like at the higher level, sort of like an umbrella. So then you can enter phenotype information, gene information. In my case, I don't have any phenotype or gene information to enter. I will add the stage. This was that I assume that was mouse at E13.5. And I think that another relevant information that I associate to this data in my example is the anatomy. And again, I'm going to click add record. And I don't know, I will say that this is a mandible angular process, mandible angular process. And as you can see, we have a very large collection basically for many of these metadata fields, and especially for anatomy. We basically have the whole Uberon ontology term. So pretty much every term in the Uberon ontology, and also all the terms in the CFA ontology and other ontologies can be found in here. So you can find in here. So I will select this one, and you can add multiple ones at this level if you need to. At this point, I see my page is starting to get a little bit confusing. So I will click here and say, okay, this is what I added so far. I have the species, stage, anatomy information. And I think that I'm kind of pretty much ready to the next part of the process, which is sort of creating the structure of your data set. By the structure of the data set, we mean how many samples have I used, what experiments I have used in my samples, and then how I will distribute the data, how I will identify and link the data that I have for each individual sample. So the first thing I will do is scroll down to the biosample section and start creating my biosamples. So in this experiment, I imagine that I basically have 2 biosamples. I will try to follow Boo's examples where he has like 1 control and 1 mutant. So I will imagine that I have 1 control sample and 1 mutant sample. So I will create a record and I will. So the first field in the biosample is what we call the local identifier. And this is one of the, probably the only free text field. And this is basically like a name. And we prefer, we've seen so that most groups are very good at and organized about how they label these samples and experiments. So if possible, and if your lab has that, we would like you to use that identifier, to follow the scheme, whatever scheme you use to identify biosamples or whatever identifier you use in your lab book. We would like you to enter that in here so that, that can be retrievable and you can find it again if you ever need to. So I don't know, I will imagine that my scheme is called lab 1, then the stage, and then there's a control. Then I will select that is mouse. The specimen, I will imagine that is a tissue specimen. In this case, I don't have any gene, genotype, strain, or mutation. But again, all these metadata fields are tied to very, pretty much all the main ontologies, like except, for example, genotype, strain, and mutation are strange ones because I think they're difficult to make coming from well-known ontologies, but they're still coming from control vocabulary. So if you want to use some genotype, for example, in this one is a control, so I will use control. But typically what happens if you are trying to add a genotype that is not in our database, then what we do is you send us an email and we add that term to the vocabulary and then you can use it. And I will assume that coming from the letter 1. And I assume that I collected it today. So here I kind of enter the main information about my sample. And now I'm going to use the clone feature to replicate that information and change only the things that are different from the previous one. So this one's going to be the mutant. And the genotype, I will change it to the LHX6 that Vu uses. And I will assume that probably comes from a different leader. And I will click save. But before I say, I want to say that this feature of replicating and cloning and adding new entries like this can be very, very helpful and we've seen people adding hundreds or even thousands of data of samples and experiments very rapidly by using this feature. So it's very good, very efficient in terms of data entry. So now I navigate back to my page. And sometimes I need to do a refresh. And now I hide the empty sections and I see that I have already created these 2 biosamples. And something that I noticed here is that I actually forgot to add the stage. And one feature that I want to show you, another very interesting feature for data entry I want to show you now is, for example, let's say I want to enter the stage field for all these biosamples. Imagine you had 10 or 20. You can click here, explore, and then do a bulk edit. And then I can go here under stage, select the 13.5 value, and I say, click apply all. And now I have populated across all my biosamples that new value. So that another feature that comes very handy when when enter a large collection of data. So now I see that, oh, and I forgot anatomy too. Okay, sorry about that. I will add again the anatomy, same way. On the process. All. Okay, I think I have everything I need. Save. Now navigate back to the dataset page. And I see that I have my 2 biosamples with all the information that I need. So the next step typically is creating the experiments. I will imagine that in this data set, I have done a micro CT analysis on these 2 biosamples, and then I've done RNX, conducting an RNA assay in, let's say, only the control biosample. So I will start by creating those 3 different experiments. So we'll tend to group them, we're trying to, if we did one micro CT on a mutant, micro CT on the control, and micro CT, and then RNA sequence, then we'll probably create 3 different experiments, but the experiments some of them can share the same sample. So I will create a record, and I will create my first experiment type, micro CT. I will enter same concept that for biosamples. If you have a sort of a scheme for labeling and giving IDs to each experiment, we would like you to enter it here. So for example, lab 1, experiment, micro CT control. And then none of these other fields, are more for the type of sequencing assay. So I can stop in here, but I can clone this one, create another experiment for the mutant, and then I can clone it one more time. But now I will change the experiment type to RNA sequencing. I will and here I think that typically we enter a little bit more information about how the sequencing has been done. And then we'll click save and navigate back to the experiment. And now we see that we have the 3 experiments and the 2 biosamples that are from my experiment, from my data set. So at this point, the only thing that it's missing is creating what we call the replicates, that are what links or associate biosamples to the experiments. And then once we create these replicates, is adding the data to the replicate. So the actual data files, the actual data is added to the replicate. So to do that, let's start by go to the first experiment, create a replicate. So here, when basically the only thing you need to do is select the biosample, and it gets filtered already by the biosample that you enter. So for this one, I will create a control biosample. Okay. Then I go back to my data set, navigate to the next experiment. Now I will create the replicate that connects my mutant biosample to my mutant microCT experiment. If you have here a large, like 3 or 4 replicates, then you will here use the clone feature here and select the different biosamples and then increment the bioreplicate number, because then you will have a replicate group. So you have bioreplicate 1, bioreplicate 2, bioreplicate 3. But in this case, I only have 1. So I will just add that one. Then I will create a replicate for the RNA sequence experiment. So I will create, in this case, I assume that I only did the RNA sequencing on the control, so I will create only one replicate. And I go back here. And when I go back to my data set, identity sections, I see that now all the sort of information that characterizes the biosamples and some of the information that characterizes the experiment have been populated in this sort of compact view of these entities. So I can see that this experiment include samples that are mouse from a stage E13.5 and with this anatomy. So that's all information that's coming from the biosample when we establish this relationship. So at this point, there's basically 2 more things that need to be done. One is add the corresponding protocols to the experiments, because like we said, we want to make sure that every experiment has an associated protocol, if it's possible. So typically when we go through the review process, we go in detail and we check that every experiment has a protocol associated. So to do that, I go back into the experiment and I will add a record and pick a protocol. So if I'm using a protocol that already has been used before, like I don't need to create a new protocol all the time if it's the same procedure I've done, my previous microCT experiments. So I will search for microCT and I will use this one of the child lab protocols that Wu has already uploaded. So I would say that's good for me. So I will select that protocol. If the protocol doesn't exist for what I did, then I will go create new and go through the process that Wu demonstrated. But in my case, I will select an existing protocol. Same thing for the other microCT. And then I will do the same for the RNA sequencing. I know that these are the ones that apply to my case, so that's why I'm picking them. But if not, you create a new one. So I will navigate back to the data set. And one thing you can notice here, if I talk, is that now the protocol section is already populated here using the information from the protocols that we added to these individual experiments. So at this point, we're pretty much done with creating the structure of the experiment. We're going to move now to the part of adding the data, the actual files, the actual data files. And there are two mechanisms by adding data to your data sets. One is through the browser, where I will demonstrate that now in here. For example, I will go to the replicate to which I want to add data. So data files, I don't know if you remember that that's a graph that Rob's showed. the data files are actually always hanging or associated to the replicate. So I navigate to the replicate, and in the replicate, I have the option of adding data of different types. It can be sequencing data, it can be process data. We typically refer to process data as data that has been obtained by processing, for example, sequencing data. So for example, sequencing data will be probably a FASTQ file, and your process data will either be a BAN or a BAN by file. And then you can add track data, for example, which another type of process data, but specifically for being displayed on the genome browser, typically weak and big weak files. Or it can be imaging data, or it can be mesh data, or we have another option here as called array data for sort of more generic type of data that can come in, in tabular form or things like that. But okay, let's say that I want to add some of my FASTQ files for this RNA sequencing experiment. Then I will click Add Record. I will enter some of the information. Let's say this was the read number 1. I will say that it was pair ended. And then in the file section, I click Select File, and you can browse to a location in your machine and select the file. You say what's the file type, the day it was submitted, and you can again use the clone feature here to do that. 1, 2, or 3 files, and let's say that the only thing that changes in this case is the read and the file. So I will select the other one here, and I can upload 2 files at the same time to that replicate. So this mechanism can be very useful and very practical when you have to upload up to, let's say, 10 files. You can do one by one, and you can rapidly upload these files. But when you have more, I don't know why, stop. Some background process might be using the server. Okay, let's give it some time. But anyway, so what I wanted to say that many times, when you have to upload more than, let's say, 10 files, we have a tool called the Riva upload tool, very similar to the one that we showed before for the bag, and actually it's distributed in the same package, in the same distribution. Maybe I have to go back here. Let's see what happened. Let's say I already uploaded one of them. To upload the next one, I will again click Add Record. Now will upload the read number 2. Will also be pair end. I will go select file, select the file from my local computer. Select the file type, which will be FASTQ in this case. Indicate when it was submitted, and then click Save. Hopefully now it will not fail. And now when I go back to my replicate, I see that there's 2 sequencing files have been uploaded to that replicate. And if I go back to the experiment that replicate belongs to, I will also see the sequencing files listed under the experiment. And if I navigate back to my data set, I will also see that data appearing in the section for the sequencing data.